Alright guys, we have section 14.4 here, going through different types of human inheritance, and this is the section for those of you who might be interested in being like a genetic counselor. When we look at these, well, we're just going to go through all different types of things that genetic counselors would have to think through. Starting with a pedigree analysis, when we look at humans, we're not able to just pick and choose who's going to mate with who, so we kind of have to look at what has already happened. So a pedigree is basically a family tree looking at a specific trait. Those of you seniors who are doing genograms right now, this would be, I think that's what they call them, this would be similar to your genogram. So I just want to go through some symbols and what different things mean. First of all, a square is going to be a male, a circle will be a female. If either the square or circle is filled in with a color, then that means that they have a specific trait that we're looking at. Also, if we have a straight line connecting two people, that means that they mated, and then the line coming down is going to lead to their offspring. We will go from oldest on the left to youngest on the right, so this would be a family with an oldest daughter and then 3 sons. The youngest son is the only one that we're showing as getting married right here. What else do we need to know? I think that's about it. When you get a pedigree, oftentimes they will not have any of the genotypes shown, any of the letters, so you kind of have to go back through and figure out what's going on. This widow's peak is the trait. It happens to be a dominant trait. Widow's peak just means it comes to a point. Their hairline comes to a point in the front versus just going all the way across. And so when we look at this, a dominant trait, those are always pretty simple as far as knowing if somebody has the gene or not, has the allele for the dominant trait or not. What's not so simple is figuring out if they are homozygous or heterozygous. So in the case of figuring out that these two people have the trait and they're heterozygous, we're looking at an offspring down here that has 2 little w's or 2 normal alleles, or non-widow's peak alleles, and so that means one of those letters had to come from each parent. So that's how we know that these are heterozygote, and you can think that way through a bunch of these. If we look at this one, this is an offspring that is a heterozygote, and the reason we know that there's a lowercase w here is because mom had a lowercase w. Okay, and that's all she could have given, so that would have come from mom, the capital W would have come from dad, and that's it. So recessive traits, the one thing I want to point out on recessive traits, over here we have attached earlobes, attached versus free. If you have an attached earlobe, that is a recessive trait, and so all the ones that have the purple fill in here, they have attached earlobes. I think I have attached earlobes, but think about yourself right now, but what do you have? Anyway, when we look at this, we introduce a term called a carrier, and anyone who carries the gene for a recessive trait but does not actually have the recessive trait, they're called a carrier. So if we look at this one right here, that is a carrier, and that is a term you're going to want to know. Carrier. Again, my penmanship is awesome on these. But anyway, a carrier, and the reason, again, they're a carrier, they have this allele, they have the attached earlobe allele there, the lowercase f, but they are not exhibiting it. But they are able to pass it on. So here we have someone with attached earlobes, someone without attached earlobes, and they're having an offspring with attached earlobes. That means that this parent had to be a carrier, otherwise they couldn't have any offspring with attached earlobes. So, I think that's it for the pedigree. If you were a genetic counselor, you would probably spend some time explaining pedigrees to people, which can be very fascinating. And with the pedigree, this follows the Mendelian genetics thing. You could do Punnett squares for people and all of that, and that would be the pedigree analysis. Okay, now we have kind of a list of some inherited disorders. There's lots of inherited disorders, but this is just a few of them, starting with some recessive disorders. Excuse me, so some recessive disorders. I'm not going to get into a huge description of these because you can just read about them in your book, but make sure you look over the cystic fibrosis. I had actually mentioned that in a previous video, but it's a recessive disorder. Sickle cell disease, look over that in your book as well. And that one, my question for you would be, just to make sure you actually read the section of the book, is sickle cell disease is extremely common for people of African descent to be carrying sickle cell disease, and it's a recessive disorder that is not beneficial. Usually non-beneficial disorders become very, very small in the population. So, but this one's really, really large in the population. And so my question is why? Why does it show up, or why do they believe it shows up so much in the general population of people of African descent? So it's an interesting one. And then Tay-Sachs, I might, if I have time here, come back to talk a little bit about it, but it does not have a specific section about Tay-Sachs, but it does discuss it within the chapter, so make sure you know what Tay-Sachs is all about. That disease. Dominant disorders. Dominant disorders, there's one, the achondroplasia, which basically is a type of dwarfism. Make sure you just look up on that one so you're familiar with it in case there's any references to it in the future chapters. And then Huntington's disease, I'm going to talk about a little bit here. Huntington's disease is a really interesting dominant disorder because it is fatal. It'll actually kill the people that get it, and typically dominant disorders that are fatal don't get passed on because if it kills you prior to you having kids, you can't pass the gene on. Well, Huntington's disease is interesting. It's a nervous disorder where your nervous system basically starts to degenerate and eventually you die. But this doesn't start until you are 35-ish, 40, 45-ish, something like that is when it'll happen. So you've already had kids. If you were going to have kids, you've already had the kids. And then you find out you have Huntington's disease and you're like, oh no, I might have passed this on to some of my kids. So one of my close friends a couple years ago, he and I we're not in touch too much anymore, but his father had Huntington's disease. And so it's a dominant disorder. His father had it. Chances are his father only had a single allele for this disease. And so that meant that my friend Phil, he had a 50% chance that he got that allele from his father. And so he had some decisions to make because he wanted to have children and start a family, and he didn't know if he had this disease. And anyway, I'll talk about it in the next slide a little bit what he had to go through. So Tay-Sachs disease, I'm not going to spend the time to explain this, but I feel it's very important for you to know. If we go back to the Tay-Sachs disease, the book describes how Tay-Sachs disease is a recessive disease at the organism level, but if you look at it as at the metabolic level, it's actually incomplete dominance. But if you look at it at the molecular level, it's actually codominant. And this is a common thing in a lot of diseases, is that it looks one way at the entire organism. If you look at it, you either have it or you don't. But then it could either be incomplete or codominant. And I'd like you to make sure you take a look into that and understand how it can be recessive and incomplete and codominant depending at what level you look at it on. So now we're going to get back to the genetic counselor idea here. Okay, now that we've talked about some different types of diseases, back to what genetic counselors might do, the things that they might be involved with, and a lot of what they're involved with is different types of testing and then counseling based on the testing. So if we first thing, we already talked about pedigree analysis. If you were a genetic counselor, you would probably walk through some pedigree analysis with people. Then the next thing that you may be involved with is if somebody wants to know whether they are a carrier or not, you could do a genetic test for that. Back to my friend who had the Huntington's disease, or thought he might have the Huntington's disease because his dad did. He would have sat down with a genetic counselor, done a pedigree analysis just to be clear on what the chances of him having Huntington's disease would be, as well as the chances of passing it on. Then the genetic test, he actually had an option here. He could get a test that would tell him for sure whether he had the recessive allele there, and whether he had that dominant allele. And he actually chose not to have the test done because he knew that if he had the test done and he found out that he does have the Huntington's disease gene, then he would be the rest of his life, every time if his fingers were numb or if he felt a little off, he'd be wondering like, oh no, is this the beginning of the end? Am I going to start to deteriorate? So he didn't want to live that way. Instead, he just wanted to not know if he had Huntington's disease, and then they also didn't want to run the risk of passing the trait on to their children, so they actually adopted kids. But that's anyway, he had to go through all that, and he had this option of the genetic test. The genetic test, they basically scan the DNA for the gene, and they can only do this for diseases where they actually know what the gene is. So that's that. Something called an amniocentesis, and then this gets a little crazy. This would be prior to having a child, so you're already pregnant, and before the child is born, you can do an amniocentesis. And what we have here with the amniocentesis is basically they take a long needle. This just sounds so horrible, but they take a long needle, stick it through the woman's stomach, through the uterus, and into the amniotic sac. And if we let me see if I can zoom in on this section here. The amnion, or the amniotic fluid, there we are. If we look at the child, sorry, I'm leaning in here. Right here, the child is surrounded by fluid. It's kind of a protective thing, but some of the cells from the child will oftentimes fall into this fluid. So they'll take a couple milliliters of this fluid, and they'll be able to get cells out of that, and they can start to do genetic tests on the cells to figure out if this child indeed has some form of whatever the genetic disease is that you're concerned with. So that's an amniocentesis. With the amniocentesis, there's kind of a delay. It's going to be a couple weeks. The cells that they do get out of the amniotic fluid, they have to grow that in a petri dish. Can take a couple weeks. Once those cells start to grow, then they can do tests on those cells to see if they've got whatever gene it is that you're looking for. Okay, so that's the amniocentesis. The other one, the next one here, is called a CVS, or chorionic villus sampling, and that's over here. The chorionic villus sampling, it's the same idea. Let's get some of the fetal cells, if we can, and then grow them, and then we can test them for genetic disorders. With this one, what they do is they take a tube and insert it through the cervix up into the uterine, or into the uterus, and then that tube is a suction tube, and they'll actually take part of the placenta, which the placenta is what connects the child to the mom, and it surrounds the child very early on. They can take, basically suck some cells off of the placenta, and those cells from the placenta would be genetically identical to the fetus. And so they will, get those cells. Those cells, the placental cells, they divide very, very rapidly, and so by the end of the day, they can actually already do the tests that they need to figure out for genetic disorders on this. So that's the CVS. Also with these, once the cells start to divide, they can do a karyotype to look for things like Down syndrome or chromosomal disorders. This one, they're showing a chromosome missing, and so this is a problem, which in Chapter 15 we'll get into what those different types of things are. So that is CVS, or chorionic villus sampling. Now, let's see what's next. Oh, you can do an ultrasound. You guys are familiar with that. They basically bounce sound waves off of the or through the mom's stomach and off the fetus, and this, they can basically get superficial things, or is there any developmental disorders that they can identify that way. So ultrasound is very non-invasive, and that's very common. They can also do something called a fetoscopy. It looks like fetoscopy, but it's fetoscopy. And on this one, they basically, again, through the cervix, they will put a fiber optic light as well as a fiber optic camera up into the uterus, and basically take pictures or get camera footage of the child, and it's another way to get a closer look at the child and get a good view of if there's any developmental disorders happening, the physical level, anatomically, if there's something not developing correctly. So those are the range of testings that can be done. And again, if you were a genetic counselor, you may not be doing the tests, but you'd be explaining the results of the tests. And if they find something in that ultrasound that isn't quite right, then you would be explaining, hey, this might be a chromosomal error, it might be something like that. Yeah, so it'd be a very interesting field to be in. It would take lots of people skills and lots of teaching skills, actually, because a lot of times you'd be teaching maybe Mendelian genetics or things like that to the parents. So that's it for this section and for this chapter, actually. And you guys have a good day.